phases. And then when it gets to the point where it has to divide for whatever reason, whatever trigger happens, it comes to the mitotic phase. The mitotic phase broken down into 4 parts. PMAT. Prophase, metaphase, anaphase, and telophase. PMAT. P-M-A-T. Our cells are eukaryotic cells. Unlike a bacteria cell, bacteria are prokaryotic. We'll see cytokinesis after the phases. So, we are either in interphase, doing what it's supposed to be doing, or when time comes to divide, entering mitotic phase, it goes through mitosis, and these are the phases of mitosis. PMAT, prophase, metaphase, anaphase, and telophase. Prophase, the nucleus disappears. Chromosomes start to become visible, so this nuclear material starts to form into the chromosomes. Metaphase is when the chromosomes line up. They're going to divide. Here's a picture too. Yeah, see it here. Chromosomes lining up. Anaphase. Now the chromosomes separate at separate. And telophase, when the chromosomes go to the end of the cell. Where is it? Right there. Now, realize this is also going to give us the opportunity, when these chromosomes come together like this, for parts of cells to break off and switch areas, switch parts. I'll show that in the video a little bit. This is how we end up with some genetic variability. This is why, for instance, nobody in the family has had red hair since, grandma and dad's side or something. And then the shuffling of the genetic material takes place. In some cases, it can shuffle incorrectly. Part of the chromosome can break off and stick onto the wrong chromosome. This can cause, not surprisingly, a type of cancer, a type of leukemia, actually. Chronic myelogenous leukemia. That is called a T922 transfer in that case. Part of chromosome 9 breaks off and transfers and sticks on a chromosome 22. So again, imagine following a recipe of how to make chocolate cake, and then somebody takes that page out of IKEA bookshelf instructions and just sticks it in the middle. It's going to end up very unusual. It's going to end up very wrong. Surprisingly, that's called the Philadelphia chromosome, T922 chromosome. The reason for that is because it tastes like cheese stick. Seriously. And of course, it's because it was discovered in Philadelphia. Anyway, the best way to do this is to memorize the phases, PMAT, prophase, metaphase, anaphase, telophase, and then picture what's happening. As the chromosomes go towards the center, then they are attached. You can see that in metaphase, so the fibers attached to the chromosomes. Then they are pulled apart in anaphase, and then in telophase, they are separated. This is also where we're going to see the cytoplasm separate, cytokinesis. During or directly after telophase, that cytoplasm divides, so we can end up with 2 identical cells. So both of these cells are going to have the same genetic material. Except, of course, like I said, in the early stages where we see it in the egg and the sperm. All right, at the meiosis stage. We've talked about cancer a little bit already. We've talked about the microorganisms. Prokaryotic cells are not going to have a nucleus. Bacteria. Bacteria can be harmful or harmless. A lot of the bacteria that we have, especially in our gastrointestinal system, can do other things, like, for instance, synthesize vitamin K. That's where we get most of our vitamin K from. But even though people call these good bacteria, what happens if that good bacteria gets into our blood? It becomes bad. Or if that good bacteria ascends up the urethra into the bladder, you end up with a urinary tract infection. So I don't like the terms good bacteria and bad bacteria. It's sort of a misnomer because it makes it sound like you want more of this bacteria, and it just sort of depends on where you're going to have it. Okay. Talk about bacteria. Here are some of the ways bacteria are described. Bacteria are described as to their shape, cocci, circles, or rods, bacilli. In this case, where the small little circles join together, diplococci, like we see in gonorrhea, Neisseria. Streptococci, staphylococci in the clusters like grapes, streptococci in chains. And you can see in this stain, it certainly look like diplococci. They are stained a purplish color. That is because most bacteria are going to fall under a category of whether or not they hold one type of a stain or if they do not hold a type of a stain. And this is simply what we call gram staining. You may have heard of this. You'll probably see it if you're around hospitals enough. Gram staining is going to be how we begin to determine what type of a bacteria that is causing a problem. So the bacteria cell wall that you see up here, will have different thicknesses of something called peptide glycan. Depending upon how much peptide glycan it has, it will determine whether or not it holds a stain or not. Long story short, what will happen is some of the bacteria will look very, very blue under a light microscope like this. That is what we call gram positive. Or they'll look very, very pink. That's what we call gram negative, where they hold on to the saccharin stain. That's something like an E. coli pink. And then from there, we just sort of follow the protocols and determine what else the bacteria can or cannot do to determine what types they are. Viruses are the bad news in a protein coat. Viruses have to reproduce in a cell. Viruses do not respond to antibiotics. Bio means life. Anti means against, right? So antibiotics pertain to drugs that are against living things. The viruses, of course, are not a living thing. Pretty interesting. Pretty interesting that the smallest particles we know that is close to a living thing, a virus, can actually kill something like, the largest whale in the ocean. I think that's interesting. The particles that are smaller than the viruses are the prions. They used to be thought of as a type of virus, but the prions are just the small strands of DNA or RNA. We talked about those a little bit already. And bovine spongiform encephalopathy, Creutzfeldt-Jacobs, and of course, my favorite, Kuru. Look at that, bad news wrapped up in a protein coat. Now, as long as things like this, that are sticking out, or these markers here, stay the same, we can do things like make a vaccine against it. But once they start to change, then our vaccine doesn't work anymore. Do something else. Fungi, this includes yeasts and molds. So when somebody talks about a fungal infection, they're not really being specific enough, but we understand what they mean. Good fungi like mushrooms are tasty. Bad fungi like candida causes thrush. It causes the yeast infections we see in females. The tinea causes athlete's foot. Tinea pedis, tinea corporis causes ringworm. You've heard of this? Ringworm, hopefully by now you know is not an actual worm. If you don't know that, ringworm is not a worm. The reason why people thought it was a worm, because it creates a little rash in a circle that's somewhat elevated. and the child will say it's itchy. I say child because they're the ones who spread this stuff. If one child has it, chances are all the child in the household have it. Tinea unguineum is what we see in the males, also known as onychomycosis. Tinea capitis, the scalp. That's actually the most common pediatric dermatophyte infection worldwide is tinea capitis. It's not cradle cap, that's different. Cradle cap is more of an eczema. What's another tinea that you've heard of? Tinea cruis. Tinea cruis. Jock itch. That's actually a yeast infection. Fungal infection. Okay, so nice examples of these. And there we see a really nice example of thrush. Grips. Chances are this is a person who is immunocompromised, because that's where we see it in an adult. We see it in children because they're by default immunocompromised. I mentioned this already, we were talking about this earlier. When you see mom carrying that baby and the binky falls out of the baby smell, and then mom picks it up, and how does she clean it off? She licks it. Sticks it in her own disgusting mouth, and gives it to the baby and says, oh, it's only my germs, right? No. Better to clean it off. Protozoas, one-celled organisms. We talked a little bit about those. Entamoeba histolytica, very common for amoebic dysentery. We saw the trypanosoma. Trypanosome we saw in Chagas disease, Trypanosoma cruzi. Trypanosoma brucei. Brucei gambins causes the African sleeping sickness. Plasmodium, we talked about this Plasmodium falciparum, Plasmodium vivax. Plasmodium malariae causes malaria. Sneaky. Hide inside a red blood cell. It's brilliant. Prophylactic antibiotics. Prophylactic meaning this is something used to prevent something else. All right. Condom is a prophylactic. It's to prevent the lifelong problem for children. She is not happy about washing her hands or something. All living organisms are made of one or more cells. For cells to carry out metabolism, they must have energy in the form of ATP. Yeah? Yeah? A lot of people understand that use energy, like when you're lifting weights, moving things around, flexing your muscles. But what's sort of hard to imagine is how energy is actually required in the release of the muscle, release in the actin modifier. We'll get to that. We'll see that later on. But it's very interesting because that's why rigor mortis happens. You don't understand that rigor mortis is the contraction of the muscles. Well, why would a muscle contract after death? And why wouldn't it relax? Well, eventually it will. Sure. But why doesn't it relax? Why does it contract? What causes that muscle to shrink up? Pretty smart, I think. All right. 2 young boys complain of stomach aches and severe diarrhea after drinking pool water because they're children. That's what happens, man. They all have infections now. Okay. Bob has a stubborn cold for 3 days. Give an antibacterial agent. He's lasted 3 days with a stubborn cold. However, he doesn't respond to treatment, the cold persists. Why? It's not bacterial, right? Here's the problem with this. The person gets sick, they feel like they have a stuffy nose, they just feel kind of miserable all around, coughing a little bit of slight fever. They take the day off of work, they make their appointment with the doctor, they drive across town, they have to circle for parking. They sit in the office, they wait for the doctor, they go to the doctor's office, the doctor very politely says, you have a viral infection. Go home, eat some chicken noodle soup, drink lots of fluids, and get plenty of rest, and not have so much stress in your life. Oh, and on the way out, make sure you pay the copay, if we didn't already collect that from you. What is this patient gonna think? Came all the way down here and I got nothing but go home and eat chicken noodle soup? And I gotta pay for this? What's gonna happen the next time that patient gets sick?
They won't go.
Well, they won't go to that doctor. Who are they gonna go to? They're gonna go to the doctor who they get an antibiotic, or at least a prescription for an antibiotic from. Because they feel like they've gotten something, right? This is why sometimes you'll go to your doctor and your doctor will say, here's a prescription for an antibiotic if it gets worse. If the fever gets worse. If you start coughing up yellow or green stuff in the next couple of days, then fill the prescription. That's what's called the business of medicine. Because, of course, doctors want to, get paid just like everybody else. Nobody wants to work for free. So sometimes patients will make a decision based on what they feel like they got from the office visit, not the actual treatment they received. So that's unfortunate. It happens a lot, though. You'll see doctors giving out prescriptions for a very benign antibiotic. I used to have a thing in the one clinic in Chicago. We have erythromycin, erythromycin in the back, doxycycline in the back, huge jugs of it. So we just dish it out there. That way, the patient can pay for it there and not have to go down to the pharmacy. We got a lot of cash patients, Medicare, Medicaid patients. And sometimes, especially with cash patients, the prescription is just too expensive. So we just give it to them that way. And then we give them a little bit of cough syrup. Basically mix up 2/3 of a liquid Benadryl, like a children's Benadryl, and 1/3 albuterol elixir, and a couple teaspoons of that, and they felt great. Of course they did. What's the albuterol going to do? It'll make them breathe a little bit better. What's the Benadryl going to do? Make them tired. Yeah. They're going to feel great. People just come in and ask me for that stuff. It's brilliant. I have my stash. No, I'm just kidding. You're called to the local nursing home for a 17-year-old female who is short of breath. Everybody has the flu. She also has the flu. Except she has a higher fever. She's got some cloudy output in her urine. And her urine is supposed to be amber to light colored, somewhat clear. Cloudy or turbid urine indicates the presence of something else. White blood cells, perhaps. And what does that mean when you see white blood cells?
An infection.
Infection. You ever drive past a bank at 3 o'clock in the afternoon, and you see, you may or may not, you see a whole bunch of police cars surrounding the bank, and the SWAT team and all the police are pointing their guns at the front door. What's happening inside of the bank?
Robbery.
It's getting robbed. You even have to see the bank robbers to know this. No, what do you see? You see all of a sudden police show up. So when you see a whole bunch of white blood cells all of a sudden show up, you don't actually have to see the pathogen. In fact, that's what you see in a lot of pus. You see a lot of white blood cells as well as pathogens and degradation products and pathogens. So when we see something like that, we have to think, there's a lot of white blood cells here. And in this case, since we see a whole lot of white blood cells in the urine should be sterile. Why would urine be sterile? It's pee. Why would it be sterile? It's filtered. The kidneys are filtering it from where? The nephrons, the filter. It's filtering blood. And blood better be sterile, or else you got a problem. So if it's sterile blood that's being filtered, it should be sterile urine that's coming out. This is indicating it's not. This is indicating another problem. What causes the flu?
Virus.
Virus. We talk about the flu, we mean influenza, right? Not to be confused with what
Birds.
we call stomach flu. Because the stomach flu is not the actual flu, right? It's more like some other agent, but it could be an acute gastritis, it could be anything from salmonella to a rotavirus. Bird flu, the birds, pig flu, swine, monkey flu, AIDS, HIV. Okay. couple of just kind of quick videos to sort of tie some of this stuff together. I'm a really big fan of animation with cell biology, basically because when I learned all this stuff, I learned it off of paper, two-dimensional, or on slides, so it required a great deal of imagination. To see it this way is absolutely amazing, because a lot more of it just makes sense suddenly. So if you ever get the chance to, YouTube different stuff, there's a lot of good videos on cell biology, where the animation is just so helpful in understanding the material. I don't know why this doesn't move. I don't know what most of that is. Nope. Glycolysis, nope. Cell cycle mitosis, yes.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells. Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. The initiating transfer RNA, carrying its amino acid, binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site.
Notice how the ribosome acts like the platform here, and everything has to be brought to it. And it has these directions right here, one strand of them. If we want to create something, all we have to do is follow those directions. And it says to create this next protein, you have to bring in the complement to C, C, and G, which of course is going to be a G, and a C. And then it'll...
The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA and new amino acids are added to the growing polypeptide chain.
It's just going to spit out this protein like
Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
See how we built this using the ribosome, but we had to bring all the pieces in. Make sense? Okay. DNA structure, I think we've seen this a little bit already. This one's very quick.
DNA is composed of 2 strands of nucleotides wound around one another in a double helix. Each nucleotide consists of a nitrogenous base along with the sugar deoxyribose and a phosphate group. If the DNA molecule unwinds, its structure resembles a ladder. Alternating sugars and phosphate groups form the sides of the ladder, and paired nitrogenous bases form the rungs. The sugar-phosphate chains of each nucleotide strand run in opposite directions. The ends of each strand are labeled according to the carbons of the sugar molecules. One end is the 3' end, and the other is the 5' end. The 3' end of one strand connects to the 5' end of the complementary strand. This anti-parallel orientation of the 2 nucleotide strands brings the nitrogenous bases into position for hydrogen bond formation. Hydrogen bonds join the 2 nucleotide strands together.
I just like the way they unravel so you can see it straight. Now here it is with the replication. Much like a zipper.
DNA replication is the process in which one double-stranded DNA molecule is used to create 2 double-stranded molecules with identical DNA sequences. For this process to occur, the 2 DNA strands are separated and the DNA replication machinery is assembled at the replication fork. The 2 DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce 2 DNA molecules with the same DNA sequence is complementary base pairing. With complementary base pairing, adenine in one strand forms hydrogen bonds only with thymine, and guanine forms hydrogen bonds only with cytosine. For example, an incoming nucleotide with a cytosine base will form hydrogen bonds with a guanine base in the template strand. A thymine, adenine, or cytosine base cannot pair with cytosine. By this mechanism, one double-stranded DNA molecule provides the information to create 2 double-stranded molecules with identical base sequences.
You see how each of the new strands is half made up of one of the original parts of the old one? Does it make sense though, if you know that C's go with G and A's go with T's? All you need is one side. If you have one side, then it just fits. Makes sense. Lysosomes. Here is the plasma membrane. Here is the outside of the cell. Here is the inside of the cell. Here is the nucleus. Here is rough endoplasmic reticulum. Here is smooth endoplasmic reticulum. Here are ribosomes, all in the cytosol. Here is the mitochondria. Here's the Golgi apparatus. Mitochondria, the lysosomes, they're budding up.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. These enzymes digest particles or cells taken into the cell by phagocytosis. They also digest old organelles such as mitochondria. The hydrolytic enzymes that degrade proteins, nucleic acids, lipids, and carbohydrates are formed in the endoplasmic reticulum and then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as defective or worn-out mitochondria. This results in the destruction and recycling of these structures.
Okay, very quickly, endocytosis, exocytosis. This will show facilitated diffusion.
Endocytosis is a process in which cells take in materials that are too large to pass through the plasma membrane. In this process, the plasma membrane surrounds and engulfs large particles. Cells use three basic types of endocytosis, depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis.
Notice how it uses part of its membrane to create a new membrane around this pathogen, so that this does not come in contact with the cytosol, the cytoplasm. Does that make sense? We want to bring the bad stuff inside, but we don't actually want to bring it into this area, because that can cause more problems.
If the material is small enough to be suspended in the extracellular fluid, such as certain proteins, the process is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle.
I think that's a better example.
Exocytosis is a process in which materials are discharged from a cell. In exocytosis, secretory vesicles migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Now again, in doing it this way, it still keeps this barrier from the inside of the cell versus the outside of the cell, by being able to fuse that membrane. We have to imagine is this membrane is a fluid membrane. It's in motion. So it is not just one solid piece, even though it looks solid, it is actually not. So things can fuse to it. This is why I didn't like their example of, I think it was a glucose transporter as facilitated diffusion. I like this example better, the receptor-mediated endocytosis. Okay, you can see the bilipid membrane here. This is the sodium-potassium pump. This is going to pump...
The sodium-potassium exchange pump is an example of an active transport process.
It's active transport. What's that mean? Requires energy. Requires energy. Remember I said anytime you hear active transport, first thing you think of is this is going to require energy. It's going to require ATP. which means if you don't have ATP, if you don't have energy, whatever this is supposed to do is not going to work.
Active transport processes can move substances against a concentration gradient, and they require energy expenditure in the form of the breakdown of ATP. The sodium-potassium exchange pump moves sodium ions out of the cell and potassium ions into the cell. The carrier protein for sodium-potassium exchange is embedded in the cell membrane. 3 sodium ions and an ATP molecule can bind to the carrier protein on the inside of the cell membrane.
Know this forever. Sodium potassium pumps are in every cell. So understand what is happening here. We have 3 sodiums that are going to move from the inside to the outside. It's going to require energy. It's going to require ATP.
The ATP is broken down to ADP and phosphate. Simultaneously, the carrier protein changes shape and the sodium ions are transported across the membrane and released on the outside. In its new conformation, the carrier protein can now bind potassium ions on the outside of the cell. 2 potassium ions move into the carrier protein from the outside of the cell membrane, and the phosphate attached to the carrier protein is released. The carrier protein changes shape again, reverting to its original conformation and delivering the potassium ions across the membrane to the inside of the cell. It is now ready to repeat the process of sodium-potassium exchange.
So are we moving things with their concentration gradient or against their concentration gradient? Why would you say that? Because it requires energy. Very simple. If it was with the concentration gradient, it wouldn't require energy. Or that's simple diffusion. If we have to move something against its gradient, we're going to be moving it uphill. If we're moving uphill against its gradient, it requires energy. Therefore, if we have something that we're moving and we require energy to move it, it must be against its concentration gradient. Good. I'm not going to go into osmosis. We understand osmosis, right? Yes, okay. Osmosis, passive movement of water across the cycle of thermal membrane from air with low soil concentration or high soil concentration. Tonicity, however, you absolutely, without a doubt, need to know. Plasma membrane, outside of the cell, inside of the cell.
Osmosis is the movement of water across a selectively permeable membrane from a compartment where the solute concentration is lower to one where it is higher. The osmotic pressure of a solution is called tonicity. In cell biology, tonicity is determined by the relative concentrations of osmotically active solutes, dissolved molecules, such as sodium chloride, NaCl, inside and outside the cell. Thus, 3 ranges of tonicity are defined for solutions with reference to cells: isotonic, also known as normal saline, hypertonic, and hypotonic. Since the relative tonicities of intracellular and extracellular fluids affect the osmotic movement of water across the plasma membrane, cells may gain or lose volume depending on the tonicity of the extracellular environment. An isotonic solution, such as normal saline, 0.9% sodium chloride, has the same concentration of osmotically active solutes as the intracellular fluid. For example, erythrocytes placed in an isotonic solution will show neither a net gain nor a net loss of water. Thus, they will maintain their normal biconcave shape. A hypertonic solution, such as 3% saline, has a higher concentration of sodium chloride than the intracellular fluid. This results in the net loss of water from the cell. Therefore, erythrocytes placed in a hypertonic solution lose water and shrivel, a process called crenation. While they lose volume, their plasma membranes have the same surface area, which accounts for their wrinkled appearance. A hypotonic solution, such as water, has a lower concentration of sodium chloride. The higher solute concentration of the intracellular fluid creates an osmotic gradient that draws water into the cell. The osmotic pressure of water entering the cell causes it to swell or even burst, lyse. Erythrocytes placed in a hypotonic solution gain water and expand until they develop leaks or rupture completely in a process called hemolysis.
A red blood cell placed in a hypertonic solution, we're talking about the solution. So the solution's going to have a greater concentration of solid particles. Now remember, water's going to want to go where there are more solid particles. So if you take a red blood cell and put the red blood cell into a hypertonic solution, the water is going to move out of the red blood cell and into the solution. That's going to cause the red blood cell to shrink, to crenate. This is what happens when you put a red blood, and listen, pay attention, this is clever stuff, man. This is what happens when you put a red blood cell into a hypertonic solution. The red blood cell shrinks, it crenates. If you put a red blood cell into a hypotonic solution, in this case, there's going to be more solid particles in the red blood cell than in the water. Water's going to do what it likes to do, move across the cyclic chromal membrane, area of low solid concentration, area of high solid concentration. It's going to move into the cell. It's going to cause the red blood cell to blow up like a balloon and then pop, or lyse. This is what happens when you put a red blood cell into a hypotonic solution. Does that make sense? This is one of those things that you need to understand forever. So at the very least, you need to memorize what happens when you put a red blood cell. This is what happens when you put a red blood cell into a hypertonic solution. It's going to crenate. Why does it shrink? Because water goes rushing out. Why does water go rushing out? There must be a lot more solid particles here. This is what happens when you put a red blood cell into a hypotonic solution. Water goes rushing into the cell. Why does water go rushing into the cell? Because there must be more solid particles in the cell relative to the solution. It's going to blow up like a water balloon and then pop. When it comes time to take a test and you're not sure, just remember what happens when you put a red blood cell into a hypertonic solution. Just remember what happens when you put a red blood cell into a hypotonic solution. If it is in an isotonic solution, what's going to happen? Nothing. Will there be movement of water? Probably. It's a fluid membrane. But any water that moves into the cell, what's going to happen? Equal amount's going to move out, right? So there'll be no net gain or net loss of water. There'll still be movement. This is the hyper and hypotonics here refer to the solution pertaining to the red blood cell. All right, I think that's all I want to show you. Oh no, there's one more. It's down here though. This is where it talks about crossing over the reproductive system.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis I, halves the number of chromosomes, and in the second division, meiosis II, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, two homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another.
You see how it gets shuffled? This is shuffling of genetic material.
In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber, and in telophase 2, along with cytokinesis, we see the formation of four genetically different haploid cells.
So in other words, one copy of chromosome 1, one copy of chromosome 1, one copy of chromosome 2. Now, what happened? We should have one copy of chromosome 1 from mom, one copy of chromosome 1 from dad, one copy of chromosome 1 from mom, one from dad, etc. What happens if we end up with one too many chromosomes? Have you heard of this?
Defect?
Yes, definitely defect. Down syndrome. This is the result of one too many chromosome number 21s. This is what we call a trisomy. So we have one copy from mom, one copy of chromosome 21 from mom, one copy of chromosome 21 from dad, and then another copy of chromosome 21. That's too many. There are other trisomies. Many of them do not. Most of them do not. Patel syndrome, trisomy 13. Edwards syndrome, trisomy 18. These are the ones you'll hear about. There's a trisomy 9, not as common. Trisomy 15, not as common. But you might hear about those. The Edwards and Patels, these have a very short life expectancy, if at all. Might be weeks, might be less than that. Klinefelter's, this is XXY. Is that a male or a female? Has a Y chromosome. We have to say male. If there's presence of a Y chromosome, we have to say it's a male. And they look pretty pretty normal. You probably wouldn't think much of them if you saw them walking down the street. They might look tall to you, kind of lanky, but they would have problems with the sperm, they'd be sterile, they could have some other conditions. I don't know, I'm sorry? No. And this question usually comes up somewhere around now. The true hermaphrodite is extremely rare. What's more common is something called pseudohermaphroditism as a result of congenital adrenal hyperplasia or acetyl hydroxylase deficiency, testicular feminization. What ends up happening is we have what's called ambiguous genitalia. It looks like it was going to be this, but then it sort of looks like it was going to be this. That's often the result of turning off these switches, starting these, and then realizing we can't continue. So we default back to this. In some cases, if there's a chromosome missing, in other words, only 45 total chromosomes, that's bad. That's called a monosomy, and they die. There's only one that survives. Have you heard of this? Have you heard of monosomies? Do you know the monosomy that survives? It's called Turner's. And it is an XO. There's an X chromosome, and then there's nothing else, which is designated as a 0 or an O. These are, of course, are they going to be male or female? Gotta be female, no Y chromosome. They're going to be sterile, short, very interesting webbed neck. Shield-shaped chest, like broad nipples, shield-shaped chest. Usually normal intelligence, otherwise sterile, otherwise normal. So you'll hear about these. I guarantee you at some point in time in your career you'll meet somebody with Turner's, or you'll hear about somebody who's having a baby with Turner's. And of course it's going to be female. Right? There's also something called XYY syndrome. And there's no other name for it, at least not that I've ever heard of. And it's interesting because it's not very common in the general population, but it is common in the prison population, XYY.
I heard that.
Yeah. It is more common. I'm sorry?
Isn't that what they link to more aggressive behavior?
Well, that's why it's interesting that we see it in the prison population more at a greater rate than in the general population, assuming that more Y chromosome equals more testosterone, equals more aggression, equals more problem with the law, et cetera, et cetera. But that's a difficult link to make, because how do you then decide, well, this person's guilty because of what they did, and this person is guilty because of a genetic defect? Once we start learning more and more about what genes do, we're going to start down a slippery path, because we will start to see that there are going to be some genetic codes that are going to say this person is more likely to be like this. This person is more likely to have this sort of disease. That causes kind of a problem. With that information, you can make decisions like, do I want to hire this person to be the CEO of our company? Or maybe join the military? Or maybe get married? So the more information we have, the more it can be kind of dangerous. This is where the medical ethics comes into play, which we are not talking about at all right now, so I'm going to stop talking about it. Interesting stuff. No. Okay. So you get an idea. I think some of this helps a little bit, right? In being able to visualize. Don't be afraid to go online. Don't be afraid to Google stuff on YouTube. There's some really great, very short animations of different cell biology and stuff going on. There's always the Khan Academy. Have you heard of this on YouTube? Khan Academy is great. It's brilliant. They really break things down very nicely. I know I'm running a few minutes behind. But I wanted to get through some of this so we can see it.
Epithelia form the surface layer of the body.
All right, you want to take a break? You want to take a quick few minutes? 3 minutes? 4 minutes?